My name's Midhat Farooqi, and I'll be talking to you today about the use of HiFi sequencing for structural variant detection in pediatric cancer. Before I dive into that, just a little bit of background. I'm a molecular pathologist, and I work at Children's Mercy Hospital in Kansas City. And one of the things that differentiates our hospital is that we have a freestanding genomic medicine center, and also have an artist on staff that goes and makes these amazing murals. So this is the one for the CPGM. And this was established back in 2011, and what made it notable historically was that it was the first to put out rapid whole genome sequencing as a diagnostic method for babies in the neonatal intensive care unit. So this is now back over 13 years ago, and it helped diagnose a lethal seizure syndrome in a baby in a matter of days, and this would have taken weeks to months otherwise. This was using short read sequencing. And then since the Genome Center has moved and is now part of our new Children's Mercy Research Institute, represented by this shiny building here. The marks that you see on the building are actually meant to represent DNA sequencing done the old way. And it's a high school student project to see if they can figure out what the mutation is that's represented on the building. The flagship kind of research project for the CMRI is Genomic Answers for Kids. And this is a large-scale pediatric rare disease genomic project, and the goal is to sequence 30,000 children with rare disease, as well as their parents and also their siblings, to create a database of ultimately 100,000 genomes focused in on the pediatric population. This began with an impetus of looking at patients who had gone through negative clinical genomic analysis, and then using advanced methods, initially methylome and then single cell, and then in 2020 really switched to doing wholesale HiFi sequencing on these patients. And to date, it's completed over 2,000 HiFi genomes, which I believe is the largest repository of pediatric rare disease HiFi genomes in the world. And my colleague, Dr. Tifo, is here today. She's the second picture down at the bottom. She'll be giving two workshops, both on one on rare disease and one on the validation of this assay later today. So just a plug for those. And before I get in trouble, not to take away anything from the other workshops, I'm sure they're all fantastic as well. One of the main things that this HiFi sequencing in these patients found initially would be something that's familiar to all of you, I'm sure. But the image on the right shows comparison of single nucleotide variant detection by HiFi sequencing having a higher sensitivity and specificity than short read whole genome sequencing, as also indicated in one of the earlier talks. And then as one of the questions asked, it is vastly superior to short read whole genome sequencing in detecting structural variants. So that's what's represented by the table on the left. Basically a comparison of short read whole genome sequencing versus HiFi sequencing for the detection of different structural variants. And there's thousands there that we are blind to if we're only doing short read. So I'm as I said, a molecular pathologist. I am mostly interested in cancer genomics, and so this ability to better detect structural variation was of keen interest to me. And this is because for any cancer, not just pediatric cancer, but that's what I am mostly going to be focusing on. The detection of structural variation is key to understanding further subtype diagnosis, as well as better informing prognosis, and also in some cases helps direct therapy. So this is an image from the World Health Organization's classification of tumors for the hematopoietic system, and this is basically the kind of reference Bible that all the pathologists use to assign diagnoses to patients. And what you can see on the right is one can start off with saying someone has acute myeloid leukemia or acute lymphoblastic leukemia, but then there are also all of these specific genetic subtypes that are further defining what these acute myeloid leukemia cases or ALL cases are. So these are largely based on the detection of structural variants. And from a clinical perspective, they are each their own entity. So if there is an acute myeloid leukemia with a translocation 15-17 and PML-RARA fusion, that is a distinct disease treated differently than an acute myeloid leukemia with a NPM1 mutation, for example. So in the clinical lab, the process to define and find the structural variants is complicated. Right now we use a whole battery of methods, starting with chromosomal karyotyping, as well as FISH, microarray, short read whole genome or whole exome sequencing or panel sequencing. And since we don't know which of these will be informative, it sort of all commences at once. And it can be challenging, not just from a cost, turnaround time, labor perspective, but also in some cases you have limited tumor material, so then triaging how much sample goes to which of these tests is another thing to consider. So we asked, could the application of HiFi sequencing improve this process? And this was a small pilot study that we did. It started with taking 11 cases of pediatric leukemia, largely because that is what we had at the time. It's the most common type of childhood cancer, and BALLs are more common, the most common of the leukemias, and then AMLs and TALLs are less frequent. But we took 5 known cases, 2 of BALL and 3 of AML, where the known is that the genetic subtype was defined by FISH, chromosomes, etc., the methods I was showing you earlier. And then we also took 6 unknown cases, and again, we know that these are BALL, or we know that this is an AML, but what we don't know is what their genetic subtype is. And all of these cases had gone through standard clinical cytogenetic and molecular testing. Again, kind of pivoting off of GA4K with trying to find answers for cases in which we had not known what the answer was. So what we did is we went to our tumor biobank and we pulled the diagnostic sample and as well as a remission sample as a paired normal, and this also, one of the cool things about the study from my perspective is we could also, if we didn't have cells preserved or frozen, it also worked using existing DNA, so we didn't need special isolation up front. We could do it off archived samples, and this also encompassed a range of different isolation types, but largely we were testing blood and bone marrow. And then they received HiFi sequencing on a Revio, 1 flow cell per sample, so 30x whole genome. And then they were aligned and phased through the human WGS pipeline, but the somatic application was different, and so we did somatic structural variant calling using a tool known as Severus. And for the Harry Potter fans, it is named after Severus Snape, not by me, but by the person who developed it. And the point of this, I'll get into more in the next slide, but it was specifically designed for somatic structural variant calling from long-read data, and it can work on a variety of inputs. But we inputted HiFi data into this, and we asked it to analyze for break-ins, and then we did a manual assessment of whether the break-in was occurring in or near a cancer gene, and whether this was clinically relevant, i.e. was it one of the genetic subtypes that was known. So this is the tool. It uses phased breakpoint graphing in order to better delineate structural variants, and it can do this at a range of variant allele frequencies, which is why it's more attuned to the somatic structural variant detection. It can also do multi-sample input, so we can use it to do paired tumor and normal analyses. It can also do tumor-only analysis if we withhold the normal BAM. And this was work done by Dr. Keskis and Dr. Kolmogorov at the NCI in collaboration with us. So the results from this pilot study were that we were able to detect the defining structural variant in all 5 of the known cases through this method. So I'll just show one example, which is from an acute myeloid leukemia case. So you can see the 2 genes involved on the left and the right. I can zoom in to show the reads basically breaking and then pairing over to the other chromosome and then vice versa. And at the kind of near the top, you can see the VCF track, and the break end is highlighted by the 2 lines in each of the locations. So this was indicative and fit with the previous detection of a RUNX1T1 fusion and a translocation 821, which again is genetic subtype defining in an AML. Also, in cases where it was not known, we were able to delineate the exact partner gene. So we had another AML case where we knew from FISH that it was NUP98 rearranged, but this is cytogenetically cryptic rearrangement. So unless you do an RNA-based fusion panel or whole genome sequencing, the exact partner gene remains undefined. And so in this scenario, we were able to say it was a NUP98 NSD1 fusion, again, subtype defining, and also do so with much better resolution because, as shown by the previous slide, we can detect it down to the nucleotide breakpoint. And then even more excitingly for us, for the 6 kind of genetically unknown cases, we were able to resolve 2 of them by this methodology. One was a case of AML and one was a case of B-ALL. And both of these are, again, kind of hallmark genetic subtype defining. One is a KMT2A rearrangement and the other is a ZNF384 rearranged case. And going back to the WHO classification, AML with a KMT2A rearrangement is its own diagnostic entity, and then B-ALL with a ZNF384 rearrangement is in the chapter of other genetic defined alterations, but again, it's its own type of B-ALL. So I can, again, show the data from this. This is showing the same data that I was showing, but now it's phased. And again, you see the breakpoints from Severus highlighted in the top track with the 2 lines, and it again is showcasing the connection from chromosome 12 to 19, and reads showing chromosome 19 and then aligning to chromosome 12 in these 2 genes. So we then asked, why were these cases not originally detected? And this was very, or particularly difficult for this first case because we had done karyotyping, we had done FISH, we had done microarray, we had done short-read exome sequencing. We could not do RNA sequencing in this case because there was not enough sample for RNA isolation. But this is something that is not necessarily known to be or cytogenetically cryptic. It can be, but there are also other cases that come through our lab that we find a KMT2A MLOT10 fusion by karyotype and FISH. But in this case, both of them were negative. And so this was a point of contention between, friendly contention between the cytogenetics lab director and I, who said, well, how do you know that the HiFi data is correct? And so we then chatted about it and decided that, well, we had only done KMT2A FISH, and that was negative, but the HiFi data was saying that it was rearranged with MLOT10. So what if her and her lab did MLOT10 FISH, and then that could be used as a secondary proof of which method was accurate. And so when she and the lab did this, the KMT2A FISH was again negative. This is a break apart probe, so you're expecting to see 2 yellow signals if nothing is rearranged, which is what was seen. And then same for MLOT10, but now instead of seeing 2 yellow signals, you see 1 yellow signal where the probes are together, but on the other chromosome, since they are split apart, then you get a separate red and green signal. So in this case, the MLOT10 FISH was positive. And then when we looked further at the HiFi data, we found that it's actually because the MLOT10 gene in that region actually breaks off from chromosome 10, and then chromosome 10 kind of stitches back together, and that piece goes and inserts into chromosome 11 where the KMT2A gene is, and that's what leads to the fusion. But because this region is only 1.5 megabases long, it separates the probes of KMT2A, but not enough to where you can tell by eye that they've actually broken apart. So a better update to this slide would be that the karyotype was cryptic, but FISH is now positive, and the HiFi data was, of course, in fact correct. This then led to now whenever KMT2A FISH is negative, the lab reflexes to MLOT10 FISH. And data has also come out from the Children's Oncology Group saying that in about 1/3 of the cases, the KMT2A MLOT10 rearrangement can be complex or cryptic and not found by traditional methods. For the other case, the B-ALL case, similarly, this is known to be cryptic on karyotype. Neither genes, ZNF384 or TCF3, are on FISH panels for ALL, not just in our lab, kind of across the US and largely across worldwide. And then it is balanced, it's a balanced rearrangement, so it's not picked up by SNP-based microarray. Our short-read exome mostly detects or is validated to do only CNV, so that's all we analyze when we look at that. And for this case, we actually had RNA, and we did perform RNA fusion testing clinically, but neither of the genes, ZNF384 or TCF3, were on this RNA fusion panel. And they're not commonly found in RNA fusion panels for ALL across, again, labs in the US, clinically, commercial labs or academic labs. So really, regardless of where we had sent it, we would not have picked this up. And so this was very encouraging to us, and we've since expanded this study. We have now done 30 cases of pediatric cancer, and not impressive number given other numbers that from the larger scale studies, but thankfully pediatric cancer is rare, and so from our institution this is a notable contribution from my perspective. And again, we did this for 26 known structurally rearranged positive cases, and then 4 known negative cases, and we expanded it also to look at solid tumors. Again, we could use archived DNA, and we didn't necessarily need banked frozen sample. However, we did have it for some of these, and so that's what we used. For the solid tumors, I would say that the more specialized DNA isolations like nanobind really help with data yield. So that is something that we are working on, and they do the same for the leukemias as well. But again, one of my favorite things about this is that we can, for the most part, use archived DNA that's already been isolated. And then you can see the range of breakdown across the different leukemia types, and then also the different types of solid tumors. We did CNS tumors and sarcomas because they are common in the pediatric population. And in summary for this, the HiFi sequencing detected the expected structural variant in 24 of the 26 known positive cases. And the 2 negative cases were ones that we were wondering whether it would be able to detect or not, because these were cases of low tumor. So they had about 20% tumor, so we were expecting the structural variant at a 10% variant allele frequency. And with a 30x genome, that's kind of right on the cusp of what you can expect to pick up reliably, since it's you would find about 3 reads. When we increased the sequencing coverage to 60x depth, then the structural variant was detected in both of these cases. And then also importantly, HiFi sequencing did not detect any structural variants in the 4 known negative cases, so known to have a driver that's maybe more a sequence variant. So it's not detecting random structural variants left and right either. And so from my perspective, 30x depth is sufficient for sequencing samples for this purpose at 30% tumor or greater. And then for future directions, we can evaluate, we're looking to do more cases. I might try to sneak in one other example in the time that I have left, but Steve Lincoln at Invitae said this at a conference a couple of years ago, and it stuck with me, that from the Tolstoy quote that all happy families are alike, but all unhappy families are unhappy in their own way. So similarly, all straightforward genetic structural variants are alike, but all complex genetic structural variants are complex in their own way. And so then here's another AML case that has a KMT2A MLL-T10 fusion, again, not detected by KMT2A FISH or karyotyping. Again, it's known to be cryptic in a set of cases. MLL-T10 FISH was positive to, again, confirm this. But in this scenario, it's a very complex rearrangement. So just as a schematic, what's happening between the normal chromosome 10 and the normal chromosome 11 is in the tumor represented in the bottom half of that figure. And you can see that one chunk is inverted, one is broken off and rearranged and also inverted, and some are not inverted. And so this is something that we really could only tease out with HiFi sequencing, and it's not detectable by short read whole genome sequencing. And again, similarly, the KMT2A probe probes end up close together so they don't act like they've broken apart, even though they are rearranged. And you need MLOT10 fish in order to see it cytogenetically. However, the cool part of this is that we had RNA for this case, and we did long-read RNA sequencing, and isoseq/Kinnex showed the KMT2A MLOT10 fusion transcript, which was super exciting. And again, even with RNA data, you can see the reads mapping from one chromosome to the next, and then vice versa. And so then this is my last slide, almost out of time. So we're hoping to do long-read RNA sequencing in parallel with DNA, and then also look at other types of somatic variants, since we haven't even touched sequence variants or copy number or methylation yet. Not enough time in the day, unfortunately. And also we're working on tool development for other biomarkers like TMB, MSI, and HRD. With that, I'd like to thank everyone involved and all of the sources who've supported us with funding, and then also the patients and families for donating their samples. And I'm out of time for questions, but happy to chat with anyone afterwards if there are any.